Hi guys, today I'm going to show you how to do confirmatory ABO blood typing using EdvoTech Kit 491. I'm Heidi Hestrick and I've been teaching Principles of Biomed for 10 years and I'm really excited about the new curriculum and this fun lab that we get to do involving all these little blood samples and figuring out what blood type they each are. At this point you should already have done the Kastle-Meyer presumptive blood test that comes in this same kit. This video is for students, and you can watch it just to see how you're going to do this lab, and then you can do it yourselves. If you're not able to do the lab this year, though, continue with the video, and I will show you the lab and then let you interpret the results. I want to say thank you so much to EdvoTech for sending me this kit and the micropipetter and the tips to do this lab so that I could show everybody how it works. In front of you, you can see everything you need to do this lab. You should have the handout, which you can print from the description of this video, 2 strips, which should fit perfectly on the handout, so you can just lay them right on there for when you add your samples, a waste beaker or cup for your used tips, a micropipette that you have set to 50 microliters. I'll have to adjust this, and the tips that go with it, and a tube rack. And in your tube rack, you should have all of the different samples that go with this. So you should have 14 tubes total, 12 blood samples, and then 2 antibody serums. The first thing that I would recommend doing is making sure that your samples are in the same order that you'll be adding them to the strips. So if you look at them, you should first have your A, which I do, and then next should be the B, then AB, O, then we should have our crime scene 1, crime scene 2, so these represent the blood that was on the edge of the table and on the floor. We should have Anna Garcia, AG, Eric Piedmont, so mine are slightly out of order. There's Eric. Then we should have Taylor Diaz, Dominique Hall, and then SG, Sam Green, and finally Dr. Opal. And then we're going to have our antibody solutions. First we'll add the A antibodies and then we'll add the B antibodies. So I'm going to set those aside for now. The first step of this lab, the way I would do it, is to add all of your blood samples to the corresponding wells. And for every blood sample, there are actually 2 wells to which you'll want to add that blood sample, and you're going to do 50 microliters at a time. Make sure that your micropipette is set to 50 and that you have on your PPE. I'm going to go ahead and get my first tip on my micropipette. Double check that I'm using the right reagent. This is A. Oh no, teacher, mine leaked. I don't have enough. Okay, so something happened to my reagent A. I'm going to get another one. Okay, I'm not sure what happened to that, but this is why as teachers we always make an extra kit, an extra set. So I went and got some more of the reagent A, and I'm going to go ahead and open it. We'll pipette this twice. Excuse my dog. Remember to only push to the first stop to get your fluid. You want to get 50 microliters and put it in the very first well, and then we don't have to switch tips because we're using the same substance and there's nothing else in there to cross-contaminate. It would be good to centrifuge first so we can get it all down to the bottom, but the exact amount isn't super important this time, so it's not a big deal if I have slightly less. I'm done with my A, so it's now going to go into my waste cup, and so is my tip. And now I'm going to do my B sample. And you're going to do this with all your samples, so I'll go ahead and time-lapse it. Just make sure you change tips each time. Isn't it nice how fast it goes when we have time lapse? So the biggest thing to do when you're doing this is just make sure that you're actually pipetting the correct sample into the right well. Triple check yourself on that to be sure. But if you make a mistake, don't panic. Just mark up your sheet. So if you'd accidentally put Taylor Diaz in EP, you could just cross out EP and put TD and just fix it on your sheet. So I have all my blood samples in now, but I don't know what type they are until I have added my antibodies. I'm going to start with my A antibody, and the directions say to add 50 microliters of A antibody to every single well across the top. So we're going to have to do that 12 times. So your measurements will be important because if you overdo it, you're going to run out of A antibody. When you do this, what I'm going to suggest is that you pipette up and down to mix, and then that you discard the tip. So I'll demonstrate this. That means we're actually going to use 24 tips total for this whole thing, 12 per row. So I have my A antibody. I'm going to pipette as much as I need into the well, and then I'm going to pipette up and down a few times just to mix it thoroughly, because you're going to get better results if the samples are thoroughly mixed. Then I'll just switch the tip, and I will do that for every other well with the A antibody. Another option is to use the same tip over and over, but do not let it touch the fluid so it doesn't get contaminated. And then go ahead and stir each well with a sterile toothpick. But I don't have toothpicks, and I kind of like mixing via pipetting. So this is the way that I'm choosing to do it. Okay, just a couple things I want to point out there. It was hard to keep track of which well I was doing. And if you get off track and you say you think that you didn't do this one yet, but then you do, and you're mixing substances together, you're going to have all kinds of contamination. So somehow coming up with a method to keep track of where you are would be really helpful. I tried to count, but even so, I was a little concerned I was going to get off. The second thing is I did actually change my pipette to 40 microliters just because I didn't want to run any risk that I would run out of the A antibodies. Just by over pipetting or something. So I went with 40 just to be conservative. It should work just as well, so you might try that as well. Now I'm going to do the B antibodies the exact same way, but along the bottom row. I'm not sure if you could tell or not, but while I was doing that, there were a couple times the antibody solution is clear, and there were a couple times where I couldn't tell for certain if I'd actually gotten any in. I did switch tips, and then I went ahead and did 40 more just in case, because it looked like maybe I hadn't grabbed any the first time. So now you have to wait about 5 minutes for agglutination. I can see some occurring already, and if you're not going to do the lab yourself, you can stick around and I'll show you results. It is important for you to understand this is a confirmatory test, so we've already done a presumptive test. The presumptive test told us the substances at the scene were maybe blood, they were probably blood, so now we're doing a confirmatory test. And there are a couple different kinds of confirmatory tests. There's the rapid stain identification, RSID, but we are doing ABO confirmatory tests. So it's going to do two things. It's going to tell us if this really is blood for sure, but it's also going to tell us if it's A, B, AB, or O type blood. So it will tell us the blood type. And there are 8 different blood types among humans. There's A positive, A negative, B positive, B negative, AB positive, AB negative, O positive, O negative. In this test, we're only telling if it's A, B, AB, or O because we didn't do the RH factor, but we could do that as part of this. This test won't tell us for sure whose blood it is. All it will tell us is the blood type, and then we can rule some people out. So for instance, I have A positive blood, but so do more than 1 in 3 people. So if we found A positive blood at a scene, does it mean it's my blood? Not necessarily. But if we found AB negative blood, that's a lot more rare. Less than 1% of people have AB negative blood. So this will let us rule some people out and then narrow down whose blood it could be. At this point, if you have the good fortune to be able to do the lab yourself, you can stop watching this video and go ahead and do the lab, and thank your teacher for the lab prep because it takes a while to set this up. If you cannot do the lab yourself, stay tuned and I will show you the results so that you can analyze them. Now that we've waited 5 minutes, what we're looking for is agglutination. Agglutination is when the A antibodies attach to the A antigens or the B antibodies attach to B antigens. And you can tell if it's agglutinated because it's kind of cloudy. The wells where we can't easily see the black outline underneath have agglutinated, or you can try putting it over letters, and if the letters disappear, it's agglutinated. But if they're still visible, then it has not agglutinated. So let's look at each sample. If we have blood type A, it has agglutinated with antibody A. And that's what should happen because type A is named for the A antigens. So the A antibodies found the A antigens and bound to them. Type B, the agglutination is a little less obvious, but definitely if you put it over the B, it is cloudy. So there is agglutination with the B antibodies for the B blood type, which is what should happen. For AB, we should see it for both because it has A and B antibodies. It's cloudier with the A, but it is cloudy with the B as well. So that's agglutination in both wells. And for O, there's no agglutination in either well. That's why it's called O blood, because there are no antibodies. So the controls serve a couple of purposes. One, it tells us our reagents are working, and they are working if it agglutinates for anti-A and A, anti-B and B, anti-A and B and AB, and nothing in O. So it tells us our reagents and our process are solid, but two, it gives us something to compare to. So now when we look at the crime scene blood, which was an unknown blood, look to see whether it agglutinated and where, and we can compare it back to the known blood types and figure out the type of blood at the scene. We can then do the same for the 5 persons of interest in Anna Garcia. For each person, look and see whether it agglutinated with A. It looks like it did for everyone except TD and EO, but everybody else it's cloudy. And then look for B, and it only agglutinated for TD and SG for B. The other ones are clear. Now you want to color in the wells on your sheet, figure out everyone's blood type, figure out the blood type at the scene, and compare it so we can rule out certain people and we can narrow down whose blood may have been at the scene. But you will not be able to answer that conclusively until we get to DNA, which is coming up in 116.